The genome, the blueprint of life. What if it isn't what we think it is? What if DNA, as described in textbooks, in scientific discovery, is just this concept modeled upon chemical debris, mathematical probability, and which is just guessing, theoretical assumptions, theory ladenness, without the true use of the scientific method, without isolating variables. The purpose of this is to raise a few questions. This will not be a comprehensive video. We will be discussing genetics, genomic theory, genetic techniques, more in depth in the Beyond Turing community. So why don't you go and check that out? It's free to join the community once it is up, but if you want to join as a paid member to get access to all of our courses, classrooms, one-on-one discussions, things like that, you can go and check out the membership options. Obviously, this discussion isn't a denial of heredity, that, I look like my dad and mom put together. Definitely, I have denied in the past that there are heritable diseases. I think diseases have 100% to do with our environment. And again, if you want to go check out that video, it will be right here. Even the Mendelian disorders seem to fall under two different interesting categories. Once the disorder has manifested, it acts differently as before it manifested. So make sure you go check out that video if you're interested in there. But I want to raise some more questions about the DNA extraction, the structure, how we came to even know about DNA. What are we looking at fundamentally here? Because most of genomics takes place in the computer. Now, I studied molecular biology as well as biochemistry, and this was largely centered around genetics. Most of my courses were genetics and looking at the fundamental methods being used in biomedical literature, germ theory, virus theory, immunology. That's what my degree was centered around. So I want to discuss a little bit about how genetic theory, how DNA was supposedly discovered, some of the methods used. If you want a more in-depth blog post on this, there's one that I'm going to be referencing today throughout the video. And so I'm going to give it one big reference point right now. It will be at the very top of the description. You can go and check that out and give it a read. It's by Tam. I'm sure many of you read it so far, but I wanted to sort of put my thoughts out on this subject as we discuss the genetic theory further. So 1869, this all began. A guy named Miescher, he wanted to extract DNA. Obviously, that wasn't his main goal. He was just doing sort of experiments. What he did was he collected pus from surgical bandages. Took this pus, chemically treated the cells, he added acid, formed a white precipitate, added alkali, added phosphoric acid, ended up with this big chemical soup. One of the things that he noticed was that this was categorically different than proteins, which are sulfur rich. He noticed that this precipitate that he created in the laboratory was rich in phosphorus. Questionably, he added phosphoric acid instead of sulfuric acid to form his precipitate. So obviously there's a little bit of question there. And basically from this experiment, believe it or not, I know it sounds like I'm bastardizing this and obviously I'm simplifying it, but this is fundamentally how he came up with nuclein. Nuclein obviously was later renamed DNA. And in this time, there was no microscopes to confirm any of this idea, right? This is all done in the test tube. Everything was not done in isolation. I mean, you can't tell me that there was some sort of isolation or control here. How do you control the addition of phosphoric acid to get this precipitate, right? Many problems come up with this methodology, and this is kind of one of the big problems in science in general, is that we claim to use a scientific method, but the second that we're in the chemistry lab, we negate all isolation and control. Because we say, oh, well, we couldn't isolate the DNA without adding acid, so how do you expect us to control this? And that is the exact reason why this is a big problem. Scientific method, if we're going to use it, we need to use it correctly. If we're going to use empiricism, we need to use it correctly. We need to isolate variables. We cannot just assume. There was already this idea developing of neo-Darwinism, right? There was already this idea that there was heritability, right? This is not a new idea, right? And it's a valid observation, but we started to get a little bit theory laden here where we're like, oh, well, there must be, some sort of substance. There must be some sort of chemical at the basis of life, because we're moving towards this mindset of materialism. So Miescher actually had this burned soup of acidified, chemically altered cellular remains collected from pus. If we're speaking of this realistically, right? Like, the problem is you put it in a scientific paper and all of a sudden we think that it's gospel and we think that it's true. But if it doesn't adhere to the scientific method, it doesn't adhere to the scientific method. So obviously this was followed up and supposedly reproduced. This guy, Hope Sellier, he took the abdomens of dogs, repeated similar steps, burned it, acidified it, all of this good stuff, big chemical soup. He got this phosphoric substance and claimed that, Miescher was a genius and that he discovered DNA or nuclein. Here lies another problem, right? Reproducibility doesn't matter if you're not actually using the scientific method in the first place. If you do 10 steps of a chemical reaction, you're going to be able to reproduce these 10 steps of this chemical reaction, but it doesn't mean that your inductions are correct. It doesn't mean that you've developed this substance, these molecules that form the basis of life, the blueprint of life. So yes, you can reproduce the methods, but that doesn't mean that the induction was correct. It doesn't mean that the experiment was correct in the first place. The big question arises is all of these steps, must have chemically altered the DNA. I mean, if you add phosphoric acid to DNA, which is supposedly so sensitive, which is a problem that will keep coming back up. DNA is said to be one of the most sensitive things that there is in our bodies, right? They say the sun, can burn your DNA damage, and DNA damage just occurs naturally as life goes on. And, what's the damage that's going to be occurring when this is in a test tube with all of these other chemicals? Obviously, we could sit here and probably name 100 different assumptions that are taking place, that some sort of nucleus was intact in the first place. Obviously, that the nuclein retained its structure, that the DNA retained its structure, that all of these chemicals didn't alter the sample whatsoever. But there's just so many questions that arise here. And I know that there's going to be a lot of resistance, and we're going to talk about that a little bit later, because it's like, well, if DNA has never actually been extracted, then what do we know about it? How do we actually know anything about DNA? And what does this mean about all of our supposedly applied sciences that have to do with DNA? But I really want to touch on a few more components, specifically the DNA components, the bases of DNA, A, T, G, and C. Obviously discussed at length in my undergraduate degree. Never questioned where they came from, how we came to this conclusion, how we isolate them. None of these things were actually discussed either. We really focus on the applied science side of things, the techniques. You don't really talk about the foundations. If you're lucky and you have a prof that really cares, you talk about the foundations in the first lecture. And if you're unlucky, they quiz you on it, because guess what? No one studies lecture 1. News flash. So between 1885 and 1901, this guy named Kossel started meddling around with DNA and trying to perpetuate the ever-evolving theory before any proof was ever actually provided to the scientific community. Only some chemical soup, which we love so much, because with chemical soup you can induce pretty much anything. Any little theory that you want, go ahead, propose it, couple it with some sort of chemical reaction, bada bing, bada boom, you got a paper, you're published. Especially in this time period, it didn't take much. When you go back and you read the papers in the 1860s, when you go into the Annales de Chimie, which I guarantee not many scientists are doing, it's very difficult to search these archives and find the papers that you want in the first place. But then when you read the papers, you're like, geez, there was nothing done. You didn't even explain the methods properly. You might have mentioned a couple of methods here and there, but really you avoided talking about, there's no IMRAD structure. You're not talking introduction, methods, results, discussion. You're not doing that, right? Anyway, this guy Kossel isolated the adenine, guanine, cytosine, thymine, and uracil, but he didn't isolate this from DNA specifically. He isolated it from other samples, right? So he used tons of different organ meats, thymus glands, different areas of organs in different animals. And he actually guanine from a bird feces, supposedly. That's where he got that one from, which is why they call it guanine, because it was from a guano or something like that. Now, his methods that he described, he used tons of harsh chemicals, including heat, which is one of the harshest things. When you heat up the body, it dies. If our bodies get up to a certain temperature, you have heat stroke and you die. So why can we heat things up in a laboratory to 100 degrees Celsius, or, and often we go way higher, and nothing happens to the supposedly blueprints of life, to the fundamental chemicals of life? They don't get altered whatsoever. He also used mercury chloride, which again disrupts DNA. Like, when you use this in controlled studies, supposedly disrupts DNA. How they measure this, according to our new developed understanding, I don't know. But if they admit it, it must have negative implications. They used phosphotungstic acid. They used silver nitrate, tons of harsh chemicals, obtained a few residues. Again, chemical byproducts. Perhaps this was reproducible, but again, there's no evidence that any of this was reproduced actually, even though reproducibility in this sense wouldn't help because you're not employing the scientific method properly. Obviously, there was no microscope available to confirm the pairing, but they did devise this idea that A binds to T and G binds to C. And then obviously Cowan highlights the arithmetic problem, that, it just simply doesn't add up. And then in comes scientific backfill to say that, oh, actually we are correct, it's just slightly off. But our theories still fit. A binds T, G binds C. Again, it's like you learn about this and you see these models in textbooks and you think, wow, we really know what we're talking about. Then you go read the fundamental papers, which are what these textbooks are based on. Like, now, people don't do these studies anymore. Like, now, people do not isolate the base pairs of DNA. Rarely do we isolate. We have this idea of what they are. We have their chemical structure. We've made these, right? We've made these in the laboratory. We've confirmed them. We have come to a scientific consensus that this is what the chemical base pairs are of DNA. And now we actually synthesize them. We actually synthesize them in a lab. Very, very rarely do we isolate them. So when we are using base pairs, like in PCR or any other genetic technique, you're not using isolated ones. You're using synthetically formed ones that were made in a laboratory. It's not lost technology like the moon landing or anything like that. We still say that we can isolate these base pairs. We still say that we can do that, but the methods are the same, right? We still use the same harsh chemicals, burning, centrifugation, etc. And the quantification and verification is just not sound. So we lyse the cells first, so we break the cells so we can obtain the nucleus where the DNA is housed. Alcohol precipitation, phenylchloroform, we spin it, we centrifuge it, and then we hydrolyse the samples. We use nucleases, obviously, to break down the nucleus so that we can extract the DNA. None of this actually alters the DNA itself, supposedly. We use formic acid, all of these chemicals, if you look them up and you see if they cause any sort of damage to cellular components, to DNA, to the molecules of life, supposedly, you're going to find some interesting stuff there. These are super harmful to people. They're super harmful to life. You don't want to go near these things. But when we do it in a test tube, supposedly all that goes away. There is no harm to life when it's in a test tube because we're looking at dead or dying or unliving matter that's in a live state, right? You're looking at cells in a petri dish, which are supposedly supposed to mimic the cells in our body. So next in our discussion, obviously, is the structure of the helix, which again is kind of funny. All of these things I took as face value in my undergrad. I never questioned it, never went and read the paper, as most scientists or doctors or whatever do. Obviously, they don't have time to do such things, to confirm these, supposedly grounded scientific discoveries. But it's so funny, right? And this happened like 50 years later, like 1950s. Rosalind Franklin, obviously, we talked about her a lot because of the big movements going on in the world. She obviously captured X-ray pictures of DNA. X-ray pictures of DNA. Say it again in your head, and you'll realize how crazy it is. Does an X-ray cause DNA damage? Isn't this something that we talk about extensively? That's why you don't want to go get too many X-rays. Or that's why we don't use X-ray technology willy-nilly, right? We only use it in when we really absolutely need it in modern medicine even, right? So she captured a picture using X-ray of DNA, sodium DNA they called it. This was extracted from the thymus gland of a calf, I'm pretty sure. Photo 51 became the absolute basis of the double helix theory. If you look at an image of this, you think, oh, okay, interesting. Yeah, you could see how they get the double helix out of it, which is pretty interesting. Even after they did 62 hours of X-ray, which had to have caused some sort of DNA damage. But you're looking at a 2D image, right? And you can understand how they got some sort of cyclical structure because it's in the form of a circle. But all it shows is these dots, these patterns. It doesn't show base pairs because these are supposedly invisible. It showed a diffraction pattern, right? So now we have to assume to try to induce some sort of theory. We need to have an assumption. So what did they assume? They assumed the angle of the helix. They thought, oh, it's probably a helix because, as Cowan again points out, and I'll link that video of his because it's phenomenal. He explains things so very well. You know, they assumed that it was a helix, a double helix, and then they assumed the angle of it. Cowan obviously uses the analogy that if you assume that an object has 4 90-degree angles, you're probably going to get a square. You know, if you assume, you're going to get an answer. So I think that's kind of an interesting point and a great analogy, because you took this 2D image, you made a major assumption, and now we just roll with it. Like, again, there's not extra proof now. Like, this is the proof. These fundamental papers are the proof. And, you know, the extraction of the DNA relies on the extraction that we discussed earlier. So they took that precipitate, right? They took that part 1 of our discussion here. They took that extraction process, used it on the calf thymus, and then took a picture of it, 62 hours with X-ray, got a 2D image, assumed an angle, bada bing, bada boom, we have DNA, the blueprint of life. These are the foundational studies. No one questions these. Now we just roll with it. Now we are in this theory-laden mindset, right? Even though you go and read the paper and Watson and Crichton sound too confident, they talk about assume and suggest and, you know, we found that without saying how. So DNA modeling, and, you know, this will come into our discussion later, not in this video, so make sure to subscribe for more. When we discuss the Human Genome Project, they didn't isolate one human genome and create a genome out of it. They took many different people, put it all together, mixed it up, put it in the computer, bada bing, bada boom, we got a sequence. Yeah, they did use Sanger sequencing. I'm definitely straw manning the argument a little bit there, so wait for that video to come out for a more logical approach to that discussion. But the idea is the same here, right? No one actually ever obtained a pure form of DNA. No one actually confirmed that we had this purity. No one actually controlled the process to make sure that we know that this is what's actually occurring in life. Regardless of whether or not DNA occurs within our living cells, whether or not we have this blueprint, this chemical blueprint that can tell us about our lives, you know, regardless of that, we don't have the ability to understand it in the laboratory, right? None of these things are controlled. It's devoid of the scientific method. Burned protein sludge, crystallized and, you know, X-rayed, using harsh chemicals to get that precipitate in the first place. You know, it all rests on pseudoscience, right? A faulty use of the scientific method. This whole genomic theory is pseudoscience because it doesn't actually adhere to the scientific method at the basis, and that's the definition of a pseudoscience. Just because now we might be able to conduct an experiment using the scientific method based on this theory doesn't confirm the initial conclusions, you know, because those conclusions are based on unverified assumptions. It doesn't make any sense, right? That's theory-ladenness at its finest. If you start with a theory and you just keep building and building and building on it, like Santa Claus. I think Santa Claus is real. I've proven him to be real. You know, I took my video camera and I took a video camera at night, midnight. He came, he gave gifts, and he left. I think Santa Claus is real. But I actually blasted Santa Claus with phosphoric acid and 62 hours of X-ray. So I know that it's the real Santa. I know that it couldn't be any other Santas, right? It's an assumption, right? I assume that this is the only possibility, that it couldn't be mom or dad because they were up sleeping. But did I confirm that finding? No, I didn't actually isolate the variable. I should have had a video camera at their door to see if they snuck out and dressed up as Santa. But now, you know, you go to the mall and you see Santa and everyone talks about Christmas and there's all these Christmas movies, but all of these Christmas movies and Santa at the mall and all these things, they absolutely do not confirm my first finding of blasting Santa with 62 hours of X-ray. You know, it doesn't confirm that I blasted Santa with all of these chemicals. So just because we can build upon a story doesn't mean that we can confirm the original conclusions. Even in another world, we still use alcohol, we still heat it up, we still use enzymes, we still use acids, we still use centrifugation for DNA extraction. Supposedly DNA is supposed to be sensitive, so obviously none of this alters that. When it's in the test tube, none of the principles to life applies apparently. Which is a huge double standard in science. Again, we're not using any controls whatsoever. It'd be really curious to see what happens when you have chemically altered cells, like when you admittedly alter the DNA of a sample to try to denature it and then run these experiments, right? None of these things have ever been done, right? But these would be really interesting experiments. What if you do it with a different biological substance, you know? But all life, they say, has DNA because, you know, you can go and run the same chemical process and get the same precipitates and then run the same methods of verification and observation, you know, and then get the same results, right? So we can use the laboratories to get the same results, but again, it's all built upon assumptions, unverifiable assumptions, uncontrolled methods, right? So reproducibility is nothing if the first paper did not adhere to the scientific method. Obviously, this is a very charged subject. DNA is very dogmatic. We have this idea in our mind that things are heritable, especially diseases and such, which is absolutely not the case. There is a certain amount of heritability, no one denies that, but it doesn't have to be DNA, right? And maybe there is DNA inside our bodies, but I feel like we don't have any claims to be able to say such a thing, right? There's no claims to say that DNA exists as this entity, right? What we have is chemically altered, burned, centrifuged, acidified precipitate. It's not life. You cannot create life from it. So I don't know what the actual application is, right? The actual application lies in forensics, paternity tests, heredity tests, GMO, CRISPR, gene editing, PCR. All of these things have so many problems with them. You know, life in the test tube is one thing. Genomics really relies on life in computers, right? When we talk about PCR, it's all computers. You know, even now, metagenomic techniques are all in computers. Heredity, all in computers. All of this is based on computers. Now, maybe there is something that you can take from, maybe a biological sample can provide some sort of understanding of one's heredity. Maybe there are things that we can understand, but it doesn't confirm these original experiments. It's theory laden. So if we want to start fresh, we need to start at the foundation. When we look at these techniques, we'll see, what are we actually observing here? What are we actually understanding here? You know, what are we actually looking at? Rather than being theory laden, relying on unverified, uncontrolled scientific experiments. Backfill is the name of the game when it comes to this discussion. When we have assumptions, unverified assumptions, and unverified conclusions, when we don't use a scientific method, things fall apart, and we have to make assumptions and we have to backfill to be able to fit our theories to our observations, because our observations are going to contradict our theories in the empirical tradition if our theories are not correct. That's why we need the backfill. When we use genetic testing to determine whether or not someone is susceptible to a disease, we use the term susceptibility because whether or not they develop the disease, we could say, oh, well, you don't got to worry about it because, you know, you were only susceptible. It wasn't actually pure confirmation. So obviously I'm not claiming that life doesn't exist. I'm not claiming that heredity doesn't exist. I do claim that genetic diseases have never been proven to exist. We don't have the observations for them. That's the big area of scientific backfill. But I do think that the scientific method should be used properly. I think that we are too deep in the test tube and we are too deep in the computers now with genomics. We're getting away from what the true study of science actually is, right? It's supposed to be the study of life, the study of cause and effects, and we need to use the scientific method properly if we are going to use it at all. Obviously, there are many inherent problems with empiricism, but if you use it in a pure Popperian fashion through falsification, it can be used properly and can be valuable. We definitely need to ask ourselves if the methods used adhere to the inductions being proposed. Are the conclusions drawn actually in reference to the methods being used? The methods need to isolate the variables being discussed. These are important questions that you need to ask when you're reading scientific papers. We're supposed to be studying life. We're supposed to be studying cause and effects. We're not supposed to be studying computers and laboratories, which is a reflection of what we're studying, obviously. What we're discussing here today is speculation, taken as fact, and built upon. Theory-latedness at its finest. Familiarize yourself with that concept. It's very important as we continue our discussion on science moving forward. If you love this discussion, you will love the Beyond Terrain community. There we are going to be discussing these things more in depth. So we are building out classrooms on all of these topics as we move forward. If you join in early, you'll be able to shape the direction of the community. If you love genetics and you want that to be the next fundamental deep dive that we do, then you are going to be able to have a say in that. So make sure that you go join. We're going to be diving deeper. We are going to be dissecting papers. We are going to be doing live video calls, Q&As, different stuff like that, really discussing this stuff in depth. Go check out the offers. There are some great things over there. And make sure to join. I think we're still going to be in a wait list, but I'm pretty sure it's going to come out in the next week or two. So be hot on your toes, sign up, check your emails. You're not going to want to miss this. It's really going to be something amazing. As for today, I hope you guys enjoyed the video. Make sure to subscribe, like, comment, do all that good stuff to help to support the channel, to get this information out, really trying to provide a lot of free content as well. And we'll continue to do so. So make sure to subscribe. That's a great way to support us too. Hope you guys enjoyed. Take care.